quickly what a normal esophagus looks like. And you can see here at the top, this would be the lumen, or where the food would pass by up here. And this is the mucosal surface. This dark line here is the basal layer, and underneath here we have some layers of muscle. So going from the basal layer to the mucosal surface, the tissue is supposed to mature. What we expect to see is that the cells become flatter and the nuclei become darker and flatter and smaller. And that is what is called normal maturation of this stratified squamous epithelium. This is a mid-esophagus biopsy, and as when you go and look at here, so we don't have any muscle layer. All we have is the stratified squamous epithelium layer here. So first of all, notice there's something different here. It doesn't look normal right away. So going from the basal layer to the mucosal surface, you're looking for normal maturation, and it's not there. When you look in higher magnification, you can see that in this abnormal tissue layer at the mucosal surface, the cells are not flattening, and the nuclei are not getting darker and flatter. The surface, this tissue is not maturing normally. And when you look at the nuclei, instead of getting smaller, more what we call pycnotic, they are actually ballooning. And some of these nuclei are suspicious that they don't even look like human nuclei to me. Like they could be a sporal cyst or something. I am not sure. But even if it is if it is a human nuclei, it is definitely not normal here. And all of these cytoplasms are cleared out, this ballooning effect. And overall, there's like a thickness here. This is an extra layer. So one might think about something called glycogenic acanthosis, which is technically a hyperplasia in which there's another tissue layer that develops, and they don't know exactly what causes it, maybe some infectious process, but however, in this case, it's not just another tissue layer. You look at this and this is like a papillary frond. And when you look closer into this frond, you can see here that there is a larva cyst here. And if this would be probably one of the best diagnostic photos or images that one could possibly find for a specific organism. I just don't know which specific organism it is, and I will explain this in a minute. But what we have here is this is inside here, they're probably like fluid and debris, and then you have all of these worm heads like sprouting out from this central core here. And when you look at the adjacent cuts to see if you can see anything better, you don't really see very much in the cut before it, and the cut after it, you can almost just kind of imagine, if you put together a 3D image of it, this here, the one in the middle here, this is a really good diagnostic photo for at least some level of family, if not the individual species level. However, the best I could find is that this is some kind of, it looks like some kind of bladder worm. We used to call them bladder worms. It's an old-fashioned term, and this is an old resource. It's oval like Cystosarcus, but it's got multiple worm heads like this one, and it doesn't really fit the picture for, either one. It's like a combination of these two. And these would be in the family of Tinea. This is a stool sample from the same patient. It's stained with Lactophenol cotton blue. And you can see on the right here, this same structure with these worm heads coming out of this central core. And this was in a mix of what looked like these other cysts. Here's some other, so you can get an idea of the abundance of them as well. And then, there was a question about whether or not this was an plant cell or an artifact of some kind of, plant cell, but it's not. You can see the worm heads coming out of it, and you also can see one head, clearly the movement of a worm head coming out of it. So these are definitely not plant cells. And then finally, this is his stomach biopsy. And lo and behold, I find this also in the stomach. This is an H&E staining of the stomach. This is definitely the same thing. I would hope that some parasitologist, pathologist somewhere in the world would be able to identify this, but I don't have access to that expert, and I have tried everything that I could possibly imagine to get access to an expert who can help identify this. This is the distal esophagus of the same patient, and I would like to show you, first of all, on low magnification, you still see an abnormal architecture, especially here with this tissue layer forming these coves with papillary fronds within. And that I just wanted to show you. And it's still has all of the same, abnormal looking nuclei but more importantly, I need to show you that all of these areas here, which are hypereosinophilic, are embedded parasitic worms. And I will show you the diagnostic. It's not diagnostic at a species level, but it is embedded in the tissue, and it is a worm. This is a long, slender worm here, and you can see the head of the worm here, and this ball. I'm not sure if this is a nematode or, and is this the, I think this would be a diagnostic feature that helps somebody who is an expert parasitologist to at least, get this down to some kind of category of possible species or family. But I also wanted to point out here that this is a hypereosinophilic tissue response, which means the tissue is more dense. It's like probably dense and harder compared to this tissue here. And there is not a lot of inflammation. I don't see any acute inflammation at all. I don't see any eosinophils. I only see maybe a few scattered lymphocytes here and there. There looks like a couple of mitoses there, maybe. And I'm not going over all of the abnormalities in this video. There are a lot of abnormalities in these biopsies. I'm trying to point out that there are parasitic worms in this tissue in order to help this person. This patient still needs help because the pathology report was negative. And this is the biopsy. These are recuts of the same exact tissue. So there was not another procedure. There was not another biopsy taken. These are cuts of the same tissue from the same biopsy. I was debating whether or not these could be eggs, but they're too small to be schistosomiasis eggs, and I believe that they are cross sections of worms, and the tissue surrounding is, again, not inflammation, but a hypereosinophilic response with clearing out of the cytoplasm, as if the worm is eating the glycogen in the cytoplasm of the cells. That must be this particular worm's food, otherwise it would not be embedding its head into the tissue. It is eating the glycogen from the cells of the tissue, and that's why these cells appear cleared out. The glycogen has been eaten out of it, I believe. So, a photo of a different cut of those 3 structures that I feel are cross sections of worms, and it's under different lighting, and you can kind of see. I almost wonder, it almost looks like another pretty important finding here to show you. This is the mid-esophagus, and these are flukes. This one you can see, this is the ventral sucker or acetabulum, and it is used to attach itself to tissue. And up here, this will be the oral sucker. This is where it eats. And this is either a different species or in a different stage of life cycle. This is the esophagus of the same patient. So I think this should also be a diagnostic image here, hoping for that parasitologist to help us out. Just quickly here, this is also the mid-esophagus, and I just want to point out how incredibly abnormal this architecture is here. Even though you can tell that the cut was split, the reason why that happened is because there is something abnormal in the tissue that has a different density, and it caused the knife to cut it at a different angle. And you can see why here. Okay, this is just another reminder of a normal esophagus. This is normal esophagus. This is not normal. And this is another cut of the distal esophagus, and I just want to also point out that this is probably one of those larva cysts. If you remember, when we looked at the different slices of the larva cyst. Okay, remember, for example, here, where we can see this clearly, this should be a diagnostic image here. But when you look at the adjacent cuts, which are only, I think, 0.3 millimeters, it's just the next cut, you see that, you might make out an outline of what, you can imagine what it looked like in 3D if you reconstructed it 3D. So what I'm saying here is that this looks like it was probably one of those. We just don't see the diagnostic piece angle of it here. More thing I need to point out in the esophagus here, this is the distal esophagus, and I found this. So look closer. That's as far as I can zoom in. But you can see that this is a hook this is a partial head of a worm, and this is a hook, and this hook has been broken off from here. So here's the other hook broken off from this piece, and here's another hook. Now I went and looked at the other cuts, the best that I have here. So it's really hard to tell still, what is this a separate worm here? Is this, I really can't really tell very well. Maybe somebody else can identify it. Okay, this is the trichrome stain of the mid-esophagus, and this is a hookworm, and it has teeth rather than plates. You can see the outline of this here, here, here. And it is a male hookworm because of the tail here. This is a photo of the stool sample of the same patient, and you're counting 1, 2, 3, 4 teeth, and this is the head of the hookworm. This is a sneak peek of the next video because it takes a lot of time to do this. So I'm going to break this up, but this is the duodenum, and there are all kinds of parasites in here, and a bunch of special stains. So that'll be in the next video.